Hi, my name is Sunil Kumar Sahu. I am from India and currently working as a research scientist in BGI research on two major projects. One is the 10,000 plant genome sequencing project, and its sub project we can say African Orphan Crop Consortium project, that is AOCC. So I am working as the main manager of this project, and I am leading our group regarding the sequencing, assembly and data analysis of the African orphan crops. So orphan crops, as from the name we can say, is the neglected crop. If you look at the calorie intake of humans, we can see only 30 species serve as the food nutritional source, among which almost 50% calories are consumed by humans from three major crops: rice, wheat and maize. So there are so many important other nutritional crops available, which are often neglected by the researchers and industries. So in this project, in this neglected crop I can actually say underutilized crop. Although they have a very good potential to become a major crop, but because of many factors they are not utilized properly. So that's why they are called as orphan crops, underutilized. Yes. So considering the tremendous increase in the human population, and there is an urgent requirement to feed this population, and it is estimated by 2050 Africa will be the third largest contributor of the world population. And in this scenario, the generating more amount of food and highly nutritious food and crops to feed those the increasing population is a major demand or challenge in front of us. And Africa has a very unique, or you can say various varieties of orphan crops which are only utilized in sub cluster of population or some local areas. And based on the previous or existing research, we can say that those crops have very high potential to become the major crop like rice, maize and you can say wheat. So in conclusion, you can say the major goal of this project, first to develop the genetic resources for this highly important and underutilized crop, and second, by using these genomic resources, more improved breeding can be developed for these neglected crops, and then it can be utilized by the local farmers to improve their productivity, to fulfill the ever increasing demand of the nutrition biosecurity. Yes. BGI is one of the leading genomic institute in the world, and it has a very big name in the field of genome sequencing, and BGI also aims to utilize this genetic information for the welfare of the mankind, as BGI tag is omics for all. And this gave BGI a very good opportunity to utilize this sequencing technology and facilities to tackle a real world problem which can be utilized for the welfare of the mankind. With this aim, BGI actually collaborated with the AOCC to instigate this project. So BGI's major task in this project is to generate the sequencing information of the samples received from the Africa, and the second is to do the bioinformatic analysis. As you know, BGI has a very solid bioinformatic facilities even to store the data and for the analysis, and by using our resources we perform all the data analysis related to starting from the genome assembly, annotation till functional annotation of all the genomes. So now AOCC, you can see, is at the beginning phase only. So we have so far sequenced 10 reference genome of African crops, among which 8 are published. The first important outcome is that we have released the genome data of 8 African crops which are freely available publicly, so anyone can freely utilize those data in their own research. Second, by sequencing the genome of these orphan crops, particularly the leguminous plant, we have identified several nitrogen fixation related gene in this crop. Second, as we know, African crops are very much tolerant to heat and drought. So we have also found several genes related to this drought tolerance and have very high potential to be utilized to improve those varieties. And third, we have also sequenced some genomes of breadfruit and jackfruit species, which have the largest fruit in the existing plant varieties. And from those sequenced genome, we have identified several genes related to starch and sucrose biosynthesis in this species. And when we tested the transcriptome data, we have identified several higher expression of the genes related to this pathway, and we hope this can be a very good resource for further breeding and improvement of these varieties. So definitely, as our other satisfied customers worldwide, AOCC is also very much satisfied with our outcomes. As you can see, we have already published 8 genomes in very top class SCI journals, and many others are in the pipeline. So yeah, I think they are very much satisfied as we are. Yes. So this is just a beginning in the era of orphan crop genomics. There is still a huge potential in the coming years. In this project, we have planned to sequence 101 highly nutritious orphan crops. In the coming years, we plan to first finish the sequencing of those 101 crops, and then the genes or the new markers we have identified from this interesting species will be utilized for further breeding or identifying some more potential marker out of this genomic data. Of course, once the sequencing is completed, we will be releasing all the data freely to public, so they can immediately start using those genomic resources for further improvement, their own research, as well as the direct application. Yes.